Okay, so ladies and gentlemen, today we are going to look at the estimation of titratable acidity, estimation of total titratable acidity in urine, and this material is adopted by from Dr. Mahesha from college. So the aim of this experiment is to estimate the titratable acidity in urine. And the principle behind this experiment is that the titratable acidity of urine is expressed in terms of the mole, a standard alkali necessary to bring the urine from its original pH to, phenolphthalein endpoint pH, which is 8.5 or 9, by the use of phenolphthalein indicator. That is the principle. So the aim or the principle of the experiment is actually trying to bring up the pH of the urine. The normal pH of the urine is 4.5. So the principle behind titratable acidity is to increase or is to bring the pH to around a pH of 8.5 to 9 using a phenolphthalein indicator. And then the total acid excreted per day under normal conditions by normal adult varies. So you see, every day we excrete acid due to metabolism of some macromolecules like metabolism of phospholipid, metabolism of nucleic acid, metabolism of proteins. We generate a lot of acid. But the amount of the acid excreted per day by humans, it varies across individuals. So it can be, the range is usually between 200 to 500 ml of 0.1 normal sodium hydroxide. But under meat diets, active gastric digestion, renal disease, fasting, and severe diabetic acidosis, values may go up to 1,500 ml. So the acidity of the urine decreased under vegetable diets. So what if you are taking vegetable diet, the acidity of the urine is usually decreased. So that is why one of the major important things that we should understand is understand that there are some diets or there are conditions that can actually lead to an increase in the total acids. So the next thing is the requirement. What and what do we need for this experiment? With 0.1, we need 0.1 normal sodium hydroxide solution. We need 1% phenolphthalein solution, and we also need potassium oxalate. And then the procedure, we are to actually pipette 25 ml of urine in a conical flask, and then we add 5 grams of freshly powdered potassium oxalate and 0.5 ml of phenolphthalein indicator solution to the flask containing the urine sample. Then you shake vigorously, and then you titrate immediately with 0.1 normal sodium hydroxide solution from every rate till a permanent ink color appears. And then you repeat the experiment thrice and take the mean of the 3 readings for the calculation. So you have to make it in a tabular form for the 3 different titrations that you are going to carry out, and then you take the average, and then you now take the volume of the acids you use. So then after that, then you come to calculate, and we are going to use this formula. X is equal to A times V divided by 25, where A is a ml of sodium hydroxide solution used. That is the amount of, or the data value you obtain from the table. Because in this case, you are titrating the acid, you are titrating base against the acid, because urine is your acid. And then after that, then the volume of urine is created in 24 hours the volume of urine excreted in 24 hours, that is 2000 ml per day under normal conditions. So that is the volume of the urine that you are going to use. But in this case, I think since I think the volume of urine excreted in 24 hours, which is actually 200 ml per day under a normal condition. So that is 2000 ml. That is what you are going to use. And then X, which is the total acidity, But I think I will advise you to just use the volume of urine you use, which is 25 ml in the conical flask. And then you now take the total acidity. So you calculate the total acidity, which is titratable acidity X. And then you report. That is all. There is nothing difficult in the experiment. Thank you. And please try to subscribe to the channel after you finish watching the video. Thank you.